Otherwise, the participant will directly join on the YouTube.
We are online.
Not audible, sir.
We are online now. I think we can start now.
Yes, sir. We can start.
A very good morning to all and one who are present here. I'm Dr. Zishan, your host, and I welcome you all in the third session of our skill enhancement course on data handling using software packages from analysis to visualization. In this lecture, I welcome Dr. Somdutta Sinha Roy, an assistant professor in the Department of Botany at Miranda House University of Delhi. Dr. Roy brings a wealth of knowledge and experience to our skill development course. With a distinguished career spanning over 2 decades, she has held significant postdoctoral research positions at the Breast Cancer Program, Department of Oncology, Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins University in the USA, and the Department of Cancer Biology and Biochemistry at Meharry Medical College, that is also in US. A prolific researcher, Dr. Roy has authored over 65 publications, and her expertise extends to curriculum development, where she has been actively involved in shaping the BSc Botany undergraduate course. Beyond her impressive academic and research contributions, Dr. Roy is deeply committed to student mentorship, serving as a facilitator for summer research projects. She is also a key feature in various research initiatives, including a Delhi University Innovation Project. Her subject specializations lie in the fascinating fields of biochemistry and ecology, a testament to her diverse interests. Please join me in giving a warm welcome to Dr. Somdutta Sinha Roy. Over to you, ma'am.
Thank you, Dr. Jishan. That was for such a wonderful introduction. I hope everybody can hear me.
You are audible.
Okay. I'll just start my presentation. I share my screen. Just a sec. Everybody can see the presentation?
Yes, ma'am.
Is
The
it
slides
visible?
are visible.
Okay. Okay. Thank you. So I welcome you all to this wonderful initiative by Zakir Hussain College on data handling using the software packages from analysis to visualization. And I have been given the job of introducing what is data, because the course says data handling, so we should know what is data and how do we collect data, and that is what I'm going to deal with today. So data is where it all begins. That's the topic that I've given to my talk, and to understand it, just a sec. What's happening? Just a sec. It's somehow not working. I'll try it again. It's not. Okay. So it's happening now. Okay. So this is a something that I found on the net a long time back, and I always use it in my presentation. In the God, everyone trusts God, whatever God you follow, but anything else anybody says has to bring the data. Because why do I say that is because this is a very common thing that happens in today's world, that a lot of advertising and a lot of politicians and other even scientific literature may be filled with such people who kind of decide what they want to say and then tell people to kind of collect data accordingly, okay, to support their view. But data is something that will never lie. Okay, it's the people who can lie, but data never lies. That is the gist of this cartoon that I've put. So this is how data is misinterpreted. A lot of times you will hear advertisements in the television, like this particular toothpaste is going to brighten up your teeth, or this particular shampoo is going to make your hair very thick and glossy. But is that true? Where is the data? So if they cannot show you the data, that means there is a possibility that they may be misinterpreting the data. They're not lying, but they're misinterpreting, like it's shown in this example, right? So if half the students think it's 3 and other half thinks is 2 plus 2 is 5, the average will again come to 4. But is it representing, giving the correct picture? No. So that is how the data can be misinterpreted. So it is very important then to know. Okay, so before beginning, I'm sure all of you know this, but I thought I'll just start from the starting. And so, what is statistics? Because that is where we are going to use our data, right? That is where that is why we are doing this. So, whatever field you are working in, whether it is humanities or sciences, whether you are a active experimental researcher, or you do your research through questionnaires and etc., we are collecting data and we are going to apply statistics on it. So it is important to know what is statistics. It is basically a field of study where we collect, organize, summarize, and analyze data. Okay, and then based on that data, we kind of draw inferences uh, about from only the part of the data that is we are collecting. Okay, so that is what statistics is. So how did it start? It started basically as a part of political science, because that's where the name, because they wanted to find out various aspects of the governance and all. So it became it was known as political arithmetic. They wanted to find out what the people are saying, how they're going to maybe vote for you and all that. So that is where it started. And then the second source was the mathematical theory of probability. Once probability was it came into being, then there was interest in games of chance among the people. Then from there, slowly the statistics had started. So what is data in statistics? Data in statistics are basically individual observation. So that observation, the measurements are taken, observations or measurements are taken on the smallest sampling unit. So smallest sampling unit, I have shown here, put here a picture of rats, like so if you are, suppose I'm studying an effect of a certain medicine on these rats, then individual, the data will be the individual rat themselves. Okay, whatever observation I'll have from individual rats is my one datum, or collection will be called the data, right? So again, what is data? Data is a collection of information obtained in a variety of ways. So you can collect data in a variety of ways, and it can be a proof of occurrence of an incident proven by scientific method, and but it is never an assumption, because I think you know that is not how the data is collected. Data is a solid, tangible, like there's a tangible proof attached to the data. Okay, so is not, data is not something that can be made just by prediction. So anybody thinking that if I have this maybe this juice of this leaf is going to make me healthier. That is an assumption. So till the time you have data to support it, you will you cannot, like, you should not be telling that to other people. Whatever you may be thinking is your choice. So why should we know types of data in statistics? So after this, I'm going to talk about types of data, and basically, I'm trying to kind of say, why we need to know what are the different types. Okay, so first one is, of course, to identify problems correctly. So if we know what type of data we are handling, it helps us to choose further methods, because after today's lecture, you will have other lectures where you will know what to do with your data. So, to choose what you need to apply to a particular problem, you need to collect a particular kind of data. Okay. So second one says to identify the right analysis tools. So every data has some unique characteristic, and to know, what tools to use. What tools am I talking about? I'm talking about statistical tools. So exactly what statistics I need to apply, will depend on the data that you have collected, and to produce, of course, the stronger results. So types of data, the first type is data by its nature. So this, couple of types I'm going to go in details, and because those are the most common one, like this one, the qualitative and quantitative data by its nature. So what is qualitative data? Qualitative data is when you are talking about, observations by your senses. So if I say it's a pink solution, or maybe it's a hot tube, I am basically, taking observation from my senses. So that constitutes the qualitative data. Now, what is a quantitative data? Whenever you're putting numbers, when you're measuring things or counting things, that is what is a quantitative data. Okay. So now quantitative data basically refers to any information that can be quantified. So basically, you in quantitative data, what you are doing is you are answering the questions of how many, how much, how often, So this, whenever you're putting a number, you are collecting the quantitative data. Okay. So once the data is collected, like I said, you have to do other things. So very popular, data analysis, tools are there, like including regression models and ANOVA and such things. So those tests basically use the quantitative data. So you need to put numbers, right? So then we come to qualitative data. Okay. So qualitative data is basically, it is descriptive in nature. So you are basically, in that case, when you're collecting qualitative data, a lot of times you're answering the question why behind the numbers. Why is this happening? Why is a particular, I'm getting a particular set of observation. Okay. So it is a very like popular method, which includes, like, thematic analysis and conversation analysis. Like a lot of humanities research, like it involves taking you are taking interviews of people, and those time you are also collecting certain amount of qualitative data. Okay. So it's a descriptive research. The qualitative approach is subjective too. It focuses on describing an action rather than measuring it. Okay. So it is more of a, what you see, what you feel, what you observe, that is what constitute a qualitative data. So qualitative data can be collected. These are the examples of qualitative data. So just a second. Sorry. So qualitative data can be like gender, religion, marital status, type of teaching approach. So all that is qualitative data. Quantitative data is, like I said, you can put numbers. So height, weight, income, group size, all these constitute a quantitative data. So it is a good research will have a balance of both qualitative and quantitative data. So observations, what you see, suppose I am doing a field-based research, what you see in the field, constitutes the qualitative data. If I have counted something, if I have observed, taken down numbers of certain things, then that constitutes the quantitative data. And a good research, like I said, is a mixture of both qualitative and quantitative data. Okay. So this is another, yes, like I said, so categorical, you can qualitative data can be taken, categorical, observations like, male, female, smoker, non-smoker. These are nominal. I'll come to these in detail later. So I'm going to like go through this here quickly. So ordinal data are like, when you are like putting groups, you're ordering things. Okay, so if I a lot of times in the hospital, if you go, you'll be asked, what is the level of the pain from in a scale of 1 to 10 maybe. So those kind of things, you have that gives you ordinal, that comes under qualitative data. So quantitative data are discrete and continuous kinds of data where you are actually taking numbers. Okay, so discrete data is basically where you are taking whole numbers. You cannot like number of limbs or number of tooth or something like that. Okay, number of seeds germinated. It cannot be a half or a decimal value. Those are discrete values. So quantitative data can be discrete or it can be continuous. Continuous examples are like, you take a weight or a length, okay, where you are expecting a decimal value. So that are continuous, okay, data. Okay. So now we come to this discrete variable quantitative data because it is important to understand. Some variables cannot be in decimals. Okay. So if you're getting even if you're averaging out, and sometimes we see the students in the class will write 2.4 seedlings germinated. 2.4 seedlings cannot germinate, right? So they are supposed to round it off for an average purpose. Okay, so it can never be, certain kind of variables can never be in decimals, and those are called discrete variables. Okay, discrete or discontinuous variables. So what are continuous variable then? In contrast, when you are taking, suppose you're calculating, taking length of my, I'm taking in a classroom length of my seedling. So they can obviously come in decimals. Okay, so that is when. So those kind of variables which can be measured in decimal are called continuous variable. Okay, and why do we say they are continuous? Because like the example which is given here, the if I have 1.5 and 1.6, I can have a continuous set of number. Like it's the example here given is 1.57. So I can have 1.55, 1.57, 1.58 between these numbers. So these are called continuous variable. Okay, because it will depend on the measuring instrument that you're using. Okay, what is the level of accuracy that you're getting? So that is why these are called continuous variables. Then we come to the second type of data. So data based on the scale of measurement, and this is kind of related to the first one. So you will have, if you are collecting the data on the basis of this, it can be a nominal data or ordinal data, ratio data, and an interval data. Okay. So what is a nominal scale? Nominal scale or nominal simply means naming. Okay. So if I'm just naming my observation, is just on naming, on the basis of naming, it is called, it is supposed to be a nominal scale data. Like the example given here, the cancer stages. You will hear a lot of time the cancer stage this, or 1 to 4, whatever stage they are given. So that depends on certain observations that are made under microscope from a sample. So now, depending on what you're observing, you are going to grade that particular cancer to stage 1 to 4. But this is only naming, so it is not going to give you anything else. So other example includes male, female, age groups. If you're given under 15, between 15 and 30, 30 to maybe 45, something like that. So when you have such thing, that is called a nominal scale. Child, adult, married, non-married, this kind of observation if you're taking. And so a lot of times when you're filling up, even maybe for this course, you'll be filling up, certain Google forms and all, and you're giving that, data to the organizers, right? So what is your, are you a student or a teacher? If you're a teacher, are you assistant professor, associate? So that kind of things are called, that is a data in itself, and that kind of data is called data on a nominal scale. Okay, so the next one is a ordinal scale. So some certain nominal scale data can be ordered in different categories. Okay, so whenever and these categories basically don't overlap. So when you have discrete categories, but you can rank them in a way, so that is called an ordinal scale. Sometimes, like, um, pollen germination, if you're doing, because I teach botany, so pollen germination you can say, okay, if it's less than this much, or it is more than this much, or it is fully germinated, something like that. So you can classify, and you can put numbers. Below this, I'm going to call it this. So you can give a rank to the germinated pollens. So that is an order. So when you can, you know, uh, put your data, collect your data in a particular order, it is called a ordinal scale. So the next one is called an interval scale. Okay, interval scale, it's a sophisticated scale, but you can get numbers directly from the measuring instrument, like a thermometer. Example given here is of temperature. So from a thermometer, you can take a reading, a number, but what happens is that the zero in this case is an arbitrary zero. Okay, so zero does not mean zero temperature, does not, whether it is Fahrenheit or Celsius, does not mean that the temperature, so to speak, the heat in the body, so to speak, is at zero, right? So, but it's an arbitrary zero that is given, and on the basis of that arbitrary zero, then you kind of have your numbers. Maybe the body temperature is this, or the temperature of water is this, the boiling water temperature is 100 degrees or whatever. Okay, so that scale in which there is no absolute zero, so to speak, that is called an interval scale. As contrast to that, then you have a ratio scale where you know the true 0 point. Okay, something like a height, weight, length, all these are ratio scale measurements when we know that what is a zero. So a zero, when suppose a seed or a pollen has not germinated, that, so there is no length of the, tube that comes out. So that is a zero is a zero. Okay, we will, whether we are looking under a microscope or using a scale to measure the seedling, germinated seedling, so we'll know if it's not there. So that is a ratio scale of measurement. So this is a collection of everything together. So no, so we know four kind of, data based on the scale of measurement, which is nominal, ordinal, interval, and ratio. Okay, so what happens? Nominal has on excuse me has only named variables. Okay, What happens? The ordinal is named, but it is ordered. So we know which one is the lowest, which one is the highest. Okay. So when we have such ordered named variables which are ordered, that those variables are called ordinals. Then we have interval where it is named, it is definitely ordered, but so there is proportional interval between the variables. So we know the interval. Okay. Then you have the ratio. Ratio, what has happened? We can, in that, in addition to everything else, it has an absolute zero. Okay. So that is a proportional scale of measurement. Okay. So, why do we need to know what kind of data we are collecting in terms of this four things? Because then it will determine what kind of, summary statistics. That's where you begin, right? What kind of summary statistics you can apply. So you will know about these things later. I'm not going to go into them. So, the highest one is the ratio scale. Here we can apply both all mean, median, mode, and coefficient of variation, everything, to know dispersion, coefficient of variation, range, standard deviation, everything can be applied to a ratio scale data. Okay. So that is why it is important to understand that what all testing can be done afterwards. What when you are going to put the data and use the data for your study, what all can be done will depend on the what scale the data has been collected. Right. Now we come. So this I'm going to kind of go quickly through this. So the next type of data is very simple. It is data based on collection process, which is primary data or a secondary data. What is a primary data? Simply put, primary data is the one that you're collecting yourself. And what is secondary data? Secondary data is somebody has already published it, or it is there in the website of some institute or something, and you are taking the data from there. That is a secondary data. Okay. So now coming in detail. So so data collected by a researcher directly from the source, like I said. So what you do with it? So what all comes under this? Data collected by a researcher working in a lab. Okay. So data collected from by questionnaires and surveys, data collected by census, data collected during market research. So all different kinds of field, when you are collecting the data yourself, that constitutes a primary data. Okay. So why, and we'll see later why it is important, because and what is now secondary data? Okay, like I said, if the data is already published or collected by somebody else and you are using it for your own work, the that is called a secondary data. And the usefulness of secondary data is because you are obviously not collecting yourself, so it's much faster and easier to access and affordable also. Sometimes, so data collected from the government websites, like I want to see what was the maybe NOx concentrations over in Delhi over a 10 years period. I can go to CPCB website and I can take out the pollutants data from there. So that data will be my secondary data that I've used. Okay. Then data collected from literature already published. So if I'm kind of using published data which is there in the published literature, whether it is in the research papers and or the books, and then I'm kind of analyzing that data, that is a secondary data. Also from newspapers, websites, articles, anything that is already published and you're using it, that will be a secondary data. Okay. So now if we compare the primary data and the secondary data, you will see that data primary data is always real time, you have collected when you are doing the work. Secondary data is a past data. So, and primary data, obviously, to do the work itself, whether you are, you're going somewhere to, conduct a survey, or you're going around, so it needs more manpower, more time. So it is more expensive, obviously, but you have a better control. So because you are doing it yourself, the control is with the researcher. So that becomes, much more. True that way. So second, not I'll not say that secondary data is not collect, but you have less control over it. But obviously, secondary data is very useful. Sometimes, like I said, I want a 10-year data. I cannot go back 10 years to collect that data, right? And wait for 10 years to do my work. So I would rather use the secondary data collected by certain government agencies to do my work, and use the data for from there. So that is how. So you decide, the kind of work that you're doing, that will kind of decide what kind of data you are using. Okay. Then we come to the fourth kind of data. So data based on collection time. So here we have an example. So this is a example of sales of certain, suppose a company, through a period from 2003 to 2006 in four different cities. Okay. So now, if you see, if I take the data, if I want to just see, what was the sales in Atlanta City for this company over a period of 4 years, I will take, like it's showing here, I'll take this column. So this is a time series data. When you have a continuous a period of time, but the data point is the same. I'm just considering one city at this point, Atlanta, and I am considering taking the data for this city only over a period of 4 years. That is a time series data. But if I want to now compare the cities, okay, all these four cities, I want to compare what was the date, the sales for the year 2003. This will constitute a cross sectional data. So, like I said, it will depend on what you are trying to study. You will collect the data, depend on that. Okay. So, time series data, this data points typically consists of successive measurements made from same source over a period of time interval. So, like tracking daily, hourly, or weekly weather data, tracking growth rates of plants and microbes, tracking changes in application performance, medical device to visualize vital signs, okay, tracking network logs, something that you are taking one measurement from one source, but over a period of time, that is a time series data. The most common one, in this case, is the daily temperature, or a temperature, how, and this, everybody recognizes this. What is this? Health monitoring. Okay, so how is your ECG? So continuous, if you go to a hospital, or you may have seen it in the movies and all, so they have a continuous monitoring of your heartbeat and all that, okay, for your vitals. So that constitutes a time series data, right? Then cross-sectional studies, what are those? Example would be like medical study examining prevalence of cancer like across. Suppose I want to see what is the prevalence of breast cancer in all the states of India. So I will be collecting from different, okay, or I want to see, so that will be a cross-sectional study. What I have to do, I'll have to, suppose I'm doing this work in 2025, so the time point will be same, but I have the data from all the states. So now that will be a cross-sectional kind of data. Okay. So again, cross-sectional studies, have, based on collection time, multiple outcomes can be reached together, and researchers can collect all variables at one time. So I may collect, whether they are smokers. I can collect the, their food habit. So but all the data I will collect which is related to my breast cancer study, but that will be at one point across a whole state number of states. The last one is data based on sources. Okay, so now this is internal data and external data. Suppose I am from Miranda House and I collect certain data regarding student performances or something from inside my institution. That is internal data. But if I go out from outside organization, other colleges, that will be an external data. Okay, so. So why are sources of data important, and why do we have to know what we are collecting and what we are doing? So because depending on, again, your problem, what is the question that you're asking, you need to kind of decide what kind of a data you want to collect. Like I said, if I just want to study, suppose, a particular disease in Delhi, and how it has changed over a period of time. Okay, I will collect a time series data. Okay, so suppose I was talking about breast cancer, I would collect the data about breast cancer from Delhi state over a period of time. Now that was my question. So initially the question decides what kind of data you want to collect. Okay, so where from you can collect, that way sources of data. So that then where from, especially the secondary data, we all know, uh, you know you can kind of collect secondary data from the already published sources. So but for primary data especially, one of the sources can be routinely kept record, especially for the medical medical research. Lot of data are sitting in in terms of routinely kept records. Remember, if you ever visit a hospital, or you have taken somebody to the hospital, you have to fill up a form that has all the details about your food habit, your whether you drink or you smoke, or other data, like whether you have a blood pressure or a sugar, everything, all that kind of data is already there. So suppose I have to you know find out certain I want to start my work regarding some medical problem. Sometimes hospital medical records are the primary source of data that you can use for initial screening and all. Okay. um, Second one is, of course, surveys. Surveys is when which is not there in the routinely kept record, and you want to find out. Then you will have to ask questions. So when you have to ask questions, suppose specific questions for your research, then those are called surveys, right? So so that can be done obviously in the hospital setting, or you can go out, like lot of population based studies. Like if you're even for health problems, lot of time when the a researcher goes to the field and he or she is asking question to the thing, those are not a data that will be sitting somewhere. Okay, so you have a specific problem. Suppose I want to find out the effect. Suppose I want to find out very common, so rhododendrons are a very common plant in Himalaya. Suppose I think there is a change in their flowering patterns. So I want to ask, go to the field and kind of ask locals if they have seen any change in the flowering time of a particular species like rhododendron. So those are something that is not available in the secondary data, papers, or some indications you may have, but you want to do specific area or something, you will need to go and ask questions. So those are kind of are known as survey. And the third one, of course, is experiments, which are something that we know that it can be some simple science people, science teachers will already know. Whatever we do in the lab, we collect data from there, right? So those actually conducting a particular evaluation, doing a particular procedure and collecting the data, that those are experiments. Like the example given here, suppose like a nurse motivating a patient. So she can, something that is not working, she's trying what is available, she's not able to get motivate her patient. Some, maybe she wants to try something else. So those are also experiments. Okay, a lot of time we do it with students also. Okay, you do this work, maybe you'll get this, so to keep them motivated. So that kind of experiments can also be called experiments. Okay. Now, we now know that we have different kinds of data, and data can be based on how it is collected, whether you can give only name to it or you can quantify it, or various things. Five different kinds of data that I talked about. So to collect data, then we need to now go to the next part. Um, Dr. Jishan, just let me know when I have to stop for the break, or can I continue? Uh, okay, okay, perfectly
But
fine.
ma'am, I think we can have a break right now because it's a different topic, na?
That's why I thought ki maybe I can
So it would be
just.
better if we will have a break right now.
Okay. So like, I'm okay because I technically can speak for 2 hours continuous, but
But
I
our
suppose
attention time is limited,
very
ma'am.
limited. Yes. So I'll stop here and I'll kind of continue. I think 5 minutes break should be okay.
Yes, ma'am. Meanwhile,
Oh,
we will take the attendance.
okay, fine. Let me know. I'm right here. Okay.
And please keep your question ready. We will allow you in the after the end of the second session.
Okay, thank you, Jishan. So in the first part, we saw, what is data and what are the different kinds of data. But to collect the data, we have to uh, it kind of links to the next part where we talked about uh, what we have to understand what we mean by a population and sample in terms of statistics. Okay, so this is an example, a very example that I've picked up. And it's a made up example, nothing like true in this. I've picked it up from somewhere. So suppose Delhi, I want to study the diastolic blood pressure of the metropolitan cities around India. And for Delhi, like because I am in Delhi, I want according 20 we are in 2025, but this data is from 2021. This was the population of Delhi, right? So is it possible for me to kind of study every individual residing in Delhi in terms of taking his or her blood pressure? Yes. Can you hear?
Yes, ma'am,
So,
you are audible.
okay. So it will not be feasible because the number is very, very high. The kind of, the effort that a researcher has to put in, to collect everything is not possible. Okay, it's going to take too much time, too much effort, too much money to do that. So now suppose somebody from I have this data that the mean pressure of the population is 78. Let us given the fact that this is a made up number. Let me first tell you that this is a made up number, obviously. Nobody has measured everybody's but suppose this is the mean. Okay, this is a supposition. how can I how will I then study? Okay, suppose take a sample size of 25%. So this was something in if you see, this is in lakhs, right? Lakhs, number is in lakhs. Now I take only 25%, and I get, when I take the average, I see my mean pressure comes out to be 67. Okay. Now I have a feeling that this may be wrong. Okay, somebody tells me, my co-researcher or supervisor, whoever tells me, ki, nahin, this doesn't, your sample size is not correct. Then I say, okay, I'll go back and I take now a sample size of 100%, 100 persons. So now I see my mean pressure is coming to be 72. This looks a little better, but somebody tells me, no, because it's in lakhs, you need to have a larger sample size. Now I take a sample size of 1,000%, and I see my mean pressure, it now comes to be 75. From this simple example, given the fact that supposed mean was 78, what do we see? We see that the increase in the sample size made my mean pressure, okay, of the sample better and better and nearer to the population. Okay, so first we have to understand then what is this sample that we are talking about. Okay, so what I already said, that if I increase my sample size, I am going nearer to the actual mean of the overall population, which was 78. Okay, this is a hypothetical example, of course, but what, because at the end, when I'm talking about a sample, what I'm trying to estimate is for the population. Okay, so that is the first premise that we have to remember, that we are talking about a sample. We are measuring things on a sample, but we are always, you know, doing it for the population. We are estimating the thing for the population. Even for, suppose we grow certain suppose I'm growing a maize or I'm growing a moong dal in my in my classroom, and I'm estimating something. So what I'm doing is I am taking my readings out of a sample that I have, but I am making those predictions or I'm saying those results for the whole population. Fine. Then, so data derived from a sample are treated statistically, and then after calculating various statistics, which you will do later on, we kind of predict. Okay, sample mean is always an estimate of the population mean. Sample standard deviation, if I'm talking about, I've grown 100 seedlings, moong dal seedlings, and then I am saying the average, suppose the height of moong dal under these conditions is 7.5 centimeters. I'm giving it for the sample that I have, but that 7.5 centimeter that I'm saying is supposed to be true for the whole population of moong dal seeds that are there. Okay, so that is why it is important to understand what is a sample and what is a population. So population is basically, like I said, if I'm talking about a Delhi population, it's the population is out of that population, we take a certain number which will be our sample. So what is a sample? Sample is a small portion of the population which is under study. So if this is Delhi, and these all colorful, cartoons which are there represent all the people that are in Delhi. Now I have taken, suppose these number 25 initially. This out of that population is my sample. So I do things, I calculate or I take measurements for this particular. So these are human figures, but this can be plants, this can be animals, this can be anything. Okay. So you have a population and then you have a sample. Sample is part of that population which you are doing your study on. Okay, so that becomes a sample. So sample, then how are you going to now, you know what are different sampling? So collecting the sample, how you collect the sample, how you take the sample will determine a lot of things. Okay, first let us understand what are the different sampling methods. Okay, so first I'm going to talk about sampling methods, then we'll come to what we are doing with that and why is it essential to kind of understand, sampling methods become so important. Okay, so sampling methods, if you look at this figure simply, you have two, one green side and one light blue side. Okay, so the green side says probability sampling methods. Second side is non-probability sampling methods. Under probability sampling methods, we have again different kinds, which include simple random sampling, stratified random sampling, cluster sampling, and systematic sampling. Under non probability sampling methods, you have convenient sampling, judgmental or purposive sampling, snowball sampling, and quota sampling. First, we'll take it step by step. We'll try to understand what are the different kinds of sampling and how you are doing that sampling. All these methods, none of them are correct or wrong or anything. They are just different sampling methods, and it depends on what, again, like I said, for data. And what are you doing with the sampling? You're basically collecting data, right? So we already know what are the different kinds of data. Now once you collect that data, you are basically sampling, uh, doing the sampling and then collecting the data from that sample that you've taken out of your population. Okay, once you will have the data from the sample, then you will progress further and use different methods of statistics and different softwares and to do the study, right? So let us start with the probability sampling methods. probability sampling or the random sampling is basically, imagine, like, when you have a fish bowl or something, and then you then, like, you pick up a chit, who's going to speak today, or classes, maybe sometimes, like, you have the chit name chits and you pick it up. So the, like, probability sampling or the simple random sampling is basically when every one in that sample, or every has, like, everyone in the population of that place population has equal chance to be picked up as a sample, or be part of a sample. Okay, so suppose I'm talking about a class. Okay, I want to decide who is going to start the presentations, maybe, right? So I have a class of 40. I will make chits of all 40, so that is my population. Now, who is going to go first, or maybe the five people who are going to you know uh, do the debate today or present today. So I have 40 name chits. Everybody's name is there in those chits, in that bowl that I have the names in. And I mix it, and I dip the hand, and I pick up random five chits. So what happened is that everybody had equal chance of being picked, and it is completely random. Okay, and everybody has 100% probability of being picked up, right? Okay, so when it, like, completely random, so that is why it is called random sampling or probability sampling. So it, every individual or member of that population has equal opportunity to be part of the sample. Okay, so that kind of sampling is called random sampling. Okay. um, So probability, sorry, this is a kind of four different kinds of random sampling. So this is what I was talking about. You have a people in the classroom, and you just pick up. So that is simple random. Then we'll go to the other one, the cluster sampling and the systematic sampling and the stratified random sampling. Okay. So this is simple random sampling, like I said. So each item of the population has equal chances of being picked. Okay, so random is not in the sense of hit or miss or haphazard, but characterized in the way they are selected. Okay, so that is something. So most of the time, when we are talking about normal distribution, it will come later on. We are talking about a sample which is done through this method. Okay, if the your sampling method is a random sampling method, you will get that kind of a normal distribution. Okay, so that is so random sampling is considered random as long as each observation that has been taken does not affect the probability of any other observation. Okay, so that is a random sampling. So, example, suppose I pick up, I have to grow 100 seeds. I have a bag of seeds, and I just dip my hand and just collect 100 seeds, right? I just, pick it up from the bag. So each, you know, uh, seed has an equal chance of being picked. I'm trying to kind of take it from a big one, and it is completely randomized. So that is called randomized sampling or random sample. Then we come to stratified random sampling. So, suppose, population under study, I'm just reading the example from here. So, it anyway can be grouped together. Okay. So, that is called and then you pick up. So, you kind of define the groups, and then you pick up the people from that group. Okay. So you're picking up particulars from a particular strata, you're picking it up randomly, but those stratas are defined. Okay, so that is called a stratified random sampling. A very easy example I can give, like, you have students, suppose, from different different parts of India in your class, and you want to find out, if you're trying to collect a data about something. Okay, so but you want to collect it state wise. So Sometimes it will happen if you go completely into random sampling. What will happen is that you may have larger representation of certain groups, but you want that each and every group should be represented. So what you do, you kind of excuse me, you kind of group them according to maybe the state. From, you have the data with you, you group them, and then you kind of, do that picking up of the chit or something. So that kind of becomes a strat. So you're basically stratifying the population, and then you are taking from each strata, you are taking certain sample, okay, so that there are equitable distribution of all the groups, okay. So that becomes a stratified random sampling. Systematic sampling, lot of time, suppose I have a list of 1,000 students and I need to pick up only 10 or maybe 100 from there. I decide, okay, I'll just go. Let me not be biased. Let me just not be like, I just want to kind of go by numbers. Okay. So I'll say I'll pick maybe the 10th and then the 20th, then the, 20th, then the 30th. So, you kind of have a pattern in the thing. Okay. So you have a whole list and you just decide, okay, this is what I am doing to make sure the whole data is the whole set that I have, I am getting from each uh, part of that data. Okay, so that is a systematic sampling. Okay, so I have a certain name, suppose, and I say, okay, 10%, uh, 20th, 30th, 40th, whatever names are there, I just go and set a pattern and I just kind of take my sample from there. Okay, so it will depend on how big your the population you're considering is, and you have put it in, it is already in a order, and then you are taking those things. Okay, so that is called a systematic sampling. Then you have something called a cluster sampling. What is a cluster sampling? Clusters are like identified um, and included in the sample based on certain things, like, I want to, like, I have a product which is only meant for children under 15 years of age. So I will not go for, the whole population. Suppose even in a school, suppose. So then I say, okay, 15 years, maybe class 9th or 10th, and that is where I want to end. So I will try to kind of collect the data, from classes under that, where I expect people to be left. So that becomes a cluster sampling. Or I say, okay, I want to just study maybe 16 to 18. So I'll just take the higher classes, 11, 12, I just take. So that is called a cluster sampling. Basically, you have identified the cluster that you want to study, and then you kind of, take the sample based on certain parameters. That is called the cluster sampling. Okay, so because why is it important? Cluster sampling is important in the places where, certain like I said, certain demographic, there are, groups has to be formed. Suppose I am making a biology book which is meant for that group. I want a biology students, or I will, if I have I'm writing a biology book, I do not want to maybe take students who are do not have biology as their subject. So I will take the, biology students of a school. So I will just consider that group, okay, or that cluster. So depending on what I am studying, what is my need, then you kind of do a different kinds of samplings. Okay, so I'll go back to this diagram that I had put earlier. So these are four kinds of, probability sampling. So simple random, everything is randomized. You have the whole thing and you just pick up at random. Okay. Then you have stratified random. Stratified random, you kind of divide your population into certain strata. Okay, here maybe they have divided into the amount of money that people are earning, and you have the whole group, and then you pick up equal number or samples from those strata depending on how big or small the numbers are. Okay, then you have clusters again. Like I said, cluster sampling is done. Once you make the sample, clusters are made, and then you pick up the sample from those yeah. And then you have systematic sample, which, like I said, I will decide, okay, I have a population of this number. Now every fifth person or fifth sample, fifth, suppose I'm doing working with plants, I can decide, okay, I have 10 trays of seedling, so I will kind of take each from each row, the first and the last, first and the last. Okay. So that way I'm going to pick up the sample from all the 10 trays that I have, the seedlings growing. So that becomes a systematic sampling. Okay. So those are, all four are called the, a part of the probability sampling. Now coming to non-probability sampling. Okay. So, in non probability or non random sampling, so what we were talking about now earlier was completely random. I was not biased as a researcher or as a person collecting the samples. I was not biased towards those things. So, I can have an order in the system, but I was not biased towards a particular thing. But sometimes, that is not what you want to do. You want to kind of choose certain samples, deliberately or not randomly. Okay, so that is extremely subjective, and it depends on the judgment of the researcher or the students who are doing it. Okay, who are doing the work. Okay, so, and this is actually needs more expertise, because here is, you are using your judgment to decide where this thing can be, this person or this thing can be part of my sample. So that judgment is completely with the researcher themselves. And so non-probability sampling is mostly used in pilot surveys. Like, you do a small survey and then you kind of, put your case forward, and then you kind of, do a larger one. So that time when you're doing a pilot survey, you want to kind of, do very quickly the study. That time you may do a non-probability sampling or a non-random sampling. Okay, so you basically, researchers use this method in studies. I'm just reading from the slide now. Studies where it is impossible to draw random probability sampling due to time or cost consideration. So this becomes a issue. When time or cost becomes an issue, sometimes you would like to go ahead and you do a non-probability or a non-random sampling. Right? Now, first of this is a convenience sampling. So this method, suppose like the example given, I want to quickly kind of survey certain customers in the mall, or somewhere in the busy street. So I can only talk to people who are passing me. So I am standing, people who are passing me are the only ones that I'm asking the question to. Okay, so that becomes, a researcher basically has no authority to select the sample elements. So you are basically asking question, or you're taking the survey only from the people who are passing by you. So so that becomes a convenient sampling. Now another example, suppose suppose I'm collecting data, some field data on plants. Okay, and I've gone to a field or a jungle where tigers are there. No, I'm not going to, nobody's going to allow me to go to the core zone when the tiger is sitting, right? So I may have to work on the buffer zone where, and that too not inside. I may have to do, whatever I'm able to see, and take down notes on plants, which trees, which I'm able to see where the paths are there. They will never allow you to go inside where the paths are not there, even with the even if you go with the, like, men with guns and all, okay, because that is not, so that becomes a convenient sampling. Okay, it is not wrong. It is because you do not have access to everything. Sometimes you have to do this kind of sampling method. Okay, then you have a judgmental or purposive sampling. Okay, so purpose, what is the purpose of the seller, along with understanding of the target audience. Okay, so suppose the example given here, suppose I want to find out how many people, thought process of the people interested in studying for their master's degree. So selection, first thing, are you doing interested in doing your master's? If you say no, do I have to ask more questions? No. I simply, if you say yes, if you're interested, because my question is, what is the thought process of the people who are who are interested in studying up for a master's? So if somebody is not interested in studying for a master's, I don't have to know their thought process, right? I don't have to know their thing. So the moment if anybody said no to me, I say okay, fine, proceed to the next person. So that is judgmental, right? That you are judging a person depending on what is your question. If it does not fit into the purview of your work, then you are not taking that, you're not taking him or her as your sample, right? So that constitutes a judgmental sampling. Then this is another very interesting way of sampling. It is called a snowball sampling. So this may not be like, for science people this may not work, but for social sciences, a lot of people are studying, how homeless people live, what are the, maybe the drug use, or maybe like some diseases like HIV or AIDS. Nobody is going to come, like, if random will never work. Okay, and even if you try the previous one, the judgmental sampling, that will also not work, because nobody is going to come and tell you if they have, right away come and tell you if they have HIV, or if they are illegal. The example given is illegal immigrants. Is somebody going to, if you go and ask a question, are they going to tell you? No. Then how? But I want to know, if suppose I'm talking about illegal immigrants or in Delhi or somebody, what I can do is, maybe if I come to know of one person, I can ask him or her, I'm not trying to get you in trouble. Your name will be secret, but I need certain data for my work. So, can you help me? Can you, maybe connect me to more people like you? Because I just want to collect data. This is nothing for anything else. Nobody is deporting you. So, but I need a data for certain, maybe medical services, or nutrition, how much nutrition related problems and such things. So then he connects me to maybe two people, those two people connect me to next four people. Okay, so finally I am able to get my sample, which is the number required by kind of, from 1 to 2, 2 to 4, 4 to 16, something like that. So that is why this kind of sampling is called a snowball sampling. Okay, so when when do you use this? You use this for when you are not will not be getting an answer right away, right? If you ask a question directly to somebody, that, say yes or no, if you have HIV or something, nobody is going to answer you that. If you're trying to find out HIV positive people and you go and start asking random people, they're not going to ask you. So you need to have the kind of sampling that you need to, where the, you kind of get in touch with the person or the victim and then collect information, and that person kind of puts you in connection with other people. So this is nothing random about it, but for certain problems this is the only way of doing the work. So that this kind of sampling, which is completely non-random, is called a snowball sampling, because this effect is called the snowball effect, right? From 1 to 2, 2 to 4, and so on. This effect is called snowball effect, and that is why this is called a snowball sampling. Next, we come to something called a quota sampling. Okay. So for quota sampling, the selection happens on the base on a preset standard. You have created a standard, and so these sample that I'm taking should have these points. Okay, so that is called a quota sampling. Okay, so and sample will have same qualities. Obviously, sample, whenever you are saying sample, it should have the same qualities about all the population. So Gallup polls are very popular polls for poll time, when you have elections and all, or certain legislations are passed. Must have heard it, Gallup polls. So there, like so you first decide which subgroup or which population interests us, and then you try to kind of collect samples from that. Okay, suppose I have I want to find out, okay, um, maybe like within India, within India during the poll time, what are the poll questions, that what is the basis of giving a vote to a particular political party or something like that. And there are groups which I know, okay, these are going to vote according to this. There are certain subgroups of population which interest me, right? Then I will try to find out, okay, this is the problem. Okay, this is the problem. Are they going to vote on the basis of such as national interest, like so this is the problem with the thing. That is when I will kind of do a quota sampling. Is there a disturbance in the thing? Getting some. Hello? There's some. There is some kind of a disturbance happening.
Ma'am, please try to remove your earphone and then speak directly. I think that will work.
Yes, can you hear me?
Yes, we can hear you, ma'am.
Okay, I'll just take it out here. It was wrong. Okay, so that is called a quota. So we basically kind of make those groups, where we want to ask questions. What, suppose I want to know how the, quota sampling is like, I have, suppose I want to see key, okay, like Delhi has got
I'm sorry for the disturbance, but you are not audible.
मैम आप अपने आप को अनम्यूट कीजिए. No, ma'am. Still it is on mute. मैम अनम्यूट कीजिए प्लीज.
I don't know how it went. Now you, I don't know how it went to mute. I have no clue. Just a sec, give me a minute. Sorry. Sorry for that. So, basically, what I was telling is that, like, for example, a quota sampling will be, suppose I want to find out, like I was saying, that in Delhi, a major issue is with, water and electricity. Okay. And I want to find out how people are going to react to certain issues. So, like, I will not aim or take the, I will kind of try to find out where the water and electricity is a problem. So regularized colonies, you will not have problems. So there's no point of including those people in the question. They will not give me the correct, uh, data. So what I am going to try to do is, I'll try to take, the I'll just consider the populations who are living in colonies which are not regularized and have problem with, running water and electricity, and then kind of take samples from there. So what you have done, you have kind of defined, where, which part of the population is going to be, part of my, like, where I can take the sample on specific attributes. Okay. Then that becomes a quota sampling. Okay. So like it says in the slide, its subsets are age, race, sex, socioeconomic levels, religion, depending on your question again. Like, what do you want to ask and what do you want to find, as a result, you kind of do this. So, non-probability sampling, please remember, obviously the best one or the correct kind of sampling is always the random sampling. That is the gold standard, that you want to do a random sampling to get the correct result. But sometimes this non-probability sampling or, are important. So suppose I do not have time, I'm just kind of giving a hypothesis, and I cannot go and kind of, do uh, the whole population or something that I need to do. So I creating a hypothesis, right? So I can do the quota sample, I can do a non-probability sampling and get data quickly, and then kind of exploratory research. And when you have the final one, the budget and time constraint. So sometimes you just do not have the budget or time to kind of do a random sampling. Like I said, the example of somebody who is collecting data from the malls, like, you just do not have enough people to kind of cover the whole, all the people coming to the mall, because, how will you catch them? So the best one is to stand at one place, and whoever is coming near you or passing by you, you ask those questions to them. So non-probability sampling also becomes very important for certain kind of work. Okay. Now, how do you decide on the type of sampling to be used? Okay. So for any research, obviously, for any kind of data collection, obviously you have to kind of choose a sampling method. And the more accurately you choose those sampling methods, more, you will be able to meet your goals of your studies. Okay. So effectiveness of your sampling obviously relies on various factors. And so the best way to go about is obviously to jot down research goals. So it is always a combination of cost, precision, and accuracy. Okay. So what is my goal? Am I, will I be able to kind of, like I said, okay, precision and accuracy, precision or accuracy is something that is always a question. How many samples are enough? How many samples I can be sure, like I showed initially, like, same thing, if you had 25 people for blood pressure or 100 or 1,000, means change. That means what? Your precision and accuracy is affected by the number that you are taking. Okay. So it becomes very, very important to jot down the research goal. What is my research? Is it something that I'm doing, like, as an initial part of a certain research project? Okay. And I'm not, like, it is not something that is going to go to the market, or it is not something, some drug that is going to directly be tested on humans. Okay. then obviously you can go with lesser number, less precision and accuracy. But at the end, when as you are going to release a product or release a drug in the market, your then you do not think about this. Your cost can go high, but precision and accuracy becomes more and more important. Okay. Then second point, identify the effective sampling techniques that might potentially achieve the research goals. Now, if I want to suppose study certain plant, and I'm able to do that work in suppose Rajasthan and Haryana, which is near to Delhi, so I can go ahead and do my work. I do not have to travel very far away. Suppose I don't have to go to maybe Arunachal or Kerala to do. If I'm getting the same work, then I'll try to kind of take the sample from there, or kind of so depending again on the research goal that you have. What kind of sampling technique you want to use. Can you go completely random, or can you uh, you can do with the stratified, okay, this is the states that I worked on these plants and this is my result. Do I have to kind of talk about all pan India, or I can just do in two states and say? So that will depending again how much money you have, how much time you have, what is the time frame that you have to finish your work. Okay, so test each of these methods and examine whether they help achieve your goal, and select the method that works best for the research. So it's always a good idea when you're starting your research to kind of write down your research goal, write down the time frame, write down the money that you have, and kind of decide on the sampling, what kind of sampling will work for your uh, goals. Okay, so you have to somewhere find that balance and then go ahead. Then what is the ideal sample size? There's a formula given by Sloven, and this is the formula which is there. And so you can use this formula to kind of calculate the ideal sample size that you need for your study. Okay, so suppose a population of 10,000, this is just an example, population of 10,000 clients with tuberculosis. Okay, so now a researcher kind of selected a sample size with a margin of error of 5%. Okay, so now desired size computed will be 385. So if that nurse kind of takes, she doesn't need to do all 10,000 clients. If she is able to get a sample size of 385, or maybe just under 400, that will suffice, and she can kind of confidently have confidence in her result that her sample size is correct, and whatever she is saying with that sample size, it is kind of true for the whole population of 10,000. Okay, now, so there is a generally trade-off between accuracy of the sample in representing the population value and cost associated with the sample size. Okay, there's always a thing. I would like to do a study and take maybe 1,000 quadrats to study my plants, but do I have am I getting am I getting the money to do that 1,000 yeah, maybe I just have money to do it in 2 days, and 2 days is not enough to take 1,000 quadrats. So what am I going to do? So I will kind of have to reduce my sample size, right? So that is always there. More, it is true that more samples you take, it is always true that you will reach nearer to the population, nearer to the population values, but uh, and you can be more confident about the results that you have, but it is not always possible. So you kind of reduce, once you reduce, taking large samples. Why do you have to reduce? Because it's extremely expensive and time consuming, so you do not have time. So a small sample is obviously less expensive and time consuming, but it is not as accurate. Like we have seen that initial, example with the blood pressure, that it is not accurate, but it is a trade-off between, how much time and money you have and how much accuracy you want. Okay. So, therefore, in situation requiring minimal error, maximum accuracy of prediction, large samples will be required. So, the best example of this, where you need large samples, definitely is when you're dealing with, drug production and such kind of study. And because larger you have, more confident you are before releasing your drug to the population, that okay, my drug is working because I've taken a big enough sample and I know my results are extremely accurate. Okay, so that is when you need that kind of investment in terms of money and time. Okay, now example of a, when you can do with a small study, TV ratings. Everybody knows that you get certain ratings for certain programs. Now, not everybody's home is, they have not put a particular, they have certain instrument which gives them the data, which channels are being switched on, which programs are being seen. So we know that, maybe all the people here, we have never had that thing in our houses. I don't know, but I'm assuming that none. So they kind of do maybe 100, 500 households, in cities or in some, yeah, and they just are happy with that kind of a thing, because in those TV ratings they do not need that kind of accuracy. They are okay with a little less accurate result, which are enough to predict what kind of programming they need to do. So it is always a trade-off between the accuracy. So there are places like medicine, and maybe releasing a certain maybe fertilizer in the market. You just need, okay, this has to work every time a person is using. So you make sure in those cases that you have big enough sample, and your sampling is very accurate and its sample is big enough to give your result that confidence. Something like TV ratings, it's okay, you can kind of do with little less accuracy, but you need, like, um, data from a small population will also, small sample size will also do. Okay. So appropriate, like I said, appropriate sample size depends on various considerations, including population variability, statistical issues, economic factors, and availability of participants. In general, larger samples will have smaller margin of error, and you can detect smaller differences between the samples. So later on, you will be talking about t-tests and ANOVAs and all that. You will see that between the sample differences, when you want to detect, sometimes if you're talking about a cancer drug or something, even if you are getting a cancer free or a situation for 3 days or 5 days, even that drug is considered good enough. So you need to find those smaller differences. Okay, so larger your sample size, you will be able to detect those between sample smaller differences. So there, like, a lot of considerations has to be there. But like, the next thing that I want to talk about is sampling error. This will also be covered later on. I just wanted to kind of talk about it a little bit. So sampling error basically includes systematic error and random error. Systematic error is when I'm collecting the sample, but I'm not designed my sampling nicely, or I've used a wrong method of sampling. Okay, so it's my error. So researcher, when the researcher is making that error in drawing a sample, or designing a study, that is a systematic error. But there are certain random errors which may pop up in studies, and so random errors do arise when you're taking your samples. Okay, so there are, just remember this later on, I'm sure that it will be covered in some other lecture. There are two kinds of error, a systematic error and a random error. Okay, so So best, like random errors is something that you cannot avoid, and a best sampling technique, best intentions also, even with best intentions, you will have certain kind of random errors arising while sampling. Okay, so this was the last, and thank you for your time and your patience. I am so I'm open for questions now. I'll try to finish as like you know
Thank
answer
you.
as well.
Thank you, ma'am. It, it was an honor to have you for sharing your invaluable insight on data, its collection types, as well as the sample and sampling. You include almost every part of these two topics, and your presentation was truly engaging and thought provoking. And we are incredibly grateful for the time and expertise you dedicated to our event. Now, we encourage all our participants to take this opportunity to ask any question or share your queries with us. You can type your question or just raise your hand and we will allow you to ask your question. Similarly, if you are connected through YouTube, you can ask your question in the comment box.
I
And
think
there are
there
some questions.
there
Can I read it for you?
Okay. So I can also read it actually. When conducting data collection in culturally diverse populations, how do we design instruments that are both valid across groups and free from measurement bias? So basically, like when you're doing, a cultural groups, the idea will be to kind of that final sample should have equal proportion. So, if you have suppose if suppose UP represents a very large group of population, and maybe Andamans have little population, and you're trying to bring them together, the ratio in the same ratio should be maintained in the total sample that you're taking. So that kind of takes care of that. And second, um,
Second is enlarged data.
in large scale data collection efforts like national surveys or censuses, what strategies are used to detect the correct data for data fabrication and falsification by field staff? See, as far as censuses is concerned, it is completely different. Censuses, they're supposed to reach everybody. Okay, that is the first thing. Okay, surveys, obviously data fabrication, like I said, data never lies. If the person who is handling that data can do something wrong, okay, and that is not come does not cannot be, somehow it cannot be really dealt with statistics per se. That is completely an ethical question. I can only hope that whoever is collecting the data has been given those guidelines how to collect the data, and the person with good ethics will do accordingly. But other than that, there is no way to statistically tackle the ethical questions. But like data, if you collect it properly, then obviously data is going to show you the correct picture. But as far as data cooking is concerned, that is a problem not only in your large data frames but also in scientific literature. You do have withdrawals and stuff like that, so there are a lot of assumptions that do happen. In case of interval scale, do we completely neglect zero? No, you do not completely neglect zero, but you kind of assign a zero. Like we have zero, like the example I gave of temperature scale, we assign a zero, and then you decide, okay, this much interval is my 10 degrees, this much interval is my 50 degrees. So that is done from a zero, which is a assumed zero. Okay, so you do not neglect the zero. Anything else?
Yeah, ma'am. I have some more question. I
Okay.
received through YouTube, so directly to my WhatsApp message. So when we perform experiment in the laboratory, is it also called sampling? This is the this is the question from one of the participant. What he was asking that when we are performing any experiment in our labs, so will
Yes.
it called sampling or
So mostly yes. What experiments we are doing in the lab? Suppose, like I said, I am from botany, so we do a lot of experiments by growing plants. So suppose I am growing, maize seedlings, and I'm growing 100 plants or something. That is going to be a sample. You are sampling. That is a sample, and you will work with those 100 seedling, and you will whatever you are saying is going to be true for maize. Like maize, you are not going and taking all the, suppose you have bought a bag also, you're not using a 1 kg seeds, right? So that 1 kg seeds you can consider as a population, but the 100 seeds that withdrawn from that bag to do your work is your sample. So yes, that is your sample.
Ma'am, second question is, beyond the techniques you discussed,
Hmm.
what is the one common biasness in data collection that often goes unnoticed, even by the experienced researcher like you? ऐसी क्या
Well,
चीज है जो अननोटिस चली जाती है बट वो बायसनेस ले आते हैं हमारे डेटा में?
biasness sometimes do creep in, even if you try to be completely random. I will say, and that is why those errors become important, and how you handle. You will learn about, handling the errors and how you can take care of those errors later. So the random errors somewhere arises because of, nothing that you are doing. Like, there were two errors. One was, if I have designed it wrong, okay, but there was an error which creeps up. So those errors are creep up because of certain inherent biasness that we have. Okay, sometimes you will see that, okay, I have seen this very commonly in the class. Suppose you are trying to locate a particular cell or trying to study. We always tell you, okay, you take the whole microscopic view, but the children will invariably go towards where they find that more. Okay, and then count ki, okay, these many were there. Okay, so complete unbiased, I think, becomes a difficulty when you're picking up the sample. Suppose I say you take 5 microscopic field, the students will invariably go to the field where pollen grains are looking nice and they're all germinated. So, it will not be unbiased. So, something like that do happen even with the best intentions of the researcher.
And I also have one question for you, ma'am. In the age of readily available big data,
Yes.
do traditional sampling techniques still hold the same importance, or
Remember,
their role are changing or evolving?
see, even if you're talking about big data, that big data is being generated somewhere. Some poor researcher is running some experiments to generate that big data. If you're talking about molecular biology, it is an age of, omics and proteomics and metabolomics and any omics that you think about. But please do remember that all that big data available is being generated by some poor chap, or boy or girl, doesn't matter, burning the night lamp in the lab somewhere. So yes, I think lab based studies are still very, very important for learning the ethics of the work. How to, like you said, if I want to remove the ethics, that has to be taught somewhere. Like, a person who can collect unbiased data is very, very important, and that comes from working in the lab, and that comes from generating the big data in itself. You are now not talking about running simply one gel and taking the data. Now you are running it in, maybe like 1000 2000 samples together and generating the data. That is the only difference. But you are using machines, you are using the tools, the computerized tools to kind of, you're not sitting and calculating t-test, which we do it in the class, but you are using a computer to do it, and which is fine. But you at the end have to kind of, generate that data, and that still is important.
थैंक यू मैम। मुझे लगता है कि हम पार्टिसिपेंट के क्वेश्चन पे जाएंगे, लेकिन वन ऑफ द कोऑर्डिनेटर डॉक्टर जिशान आल्सो हैव क्वेश्चन फॉर यू that right now we are moving to the FIUP, means 4-year undergraduate program in University of Delhi. So many of them will require data for their dissertation, for their research. So can you suggest some website, because it's not possible or feasible to conduct the wet laboratory experiment for everyone. So can you suggest some sources from there they can collect the secondary data and can work on this?
See, the one that I told, I think the environment related data is, a lot of it is available in the government websites. And I'm sure even the MolBio, PubMed and all has a lot of data per se. So depending on the work that you want the students to do, nowadays, because every institute and or the researcher who is using public funding is supposed to deposit the data. Yeah, and I'm sure all of you have faced that when you submit a paper you're expected to kind of release the data, so that is available. Like even for the they will just show you the graphs and the tables, but the data is available if you dig enough. The science and the good journals will always like have this data attached to it, so you can mine it that way also. But there are websites, nothing is coming off my head except CPCB and maybe PubMed and all that for that matter now. But there are data available, and where you can go and kind of use that data to do your work. So we are all in the same boat, ma'am. So I will also start looking up when I have to do it. But yes, I'm sure that life is going to get very exciting, and we will kind of have to look into secondary data sources which can be used, because, like you said, it's not going to be possible to do it with the primary data all the time. We do not have the space and the money to do certain work, so
Yes.
we will have to look.
And I'm
Yes.
sure Jishan sir also has some of the sources, so I will get back to him later on. First,
Yes.
there are 2, 3 more questions for you, ma'am, and then I will ask Jishan sir if he has some information from where
Yes.
we can
Yes.
collect.
Yes. Or I
So,
can even, if you write to me, I can even reply in my email. But off the head, I really don't remember right now.
no
I'm
worries.
sure there are data sets available.
So, there is one more question from Atif Siddiqui. Can the act of collecting data itself change the behavior of the participant, and if so, how should researchers adapt their design to minimize this effect?
Mm, like you're saying, when you're collecting the data, the behavior can change depending on what questions you are asking. So that is why those especially the social sciences, where you are asking questions, sciences, we are happy because our plants and our cells do not kind of react that way, right? So they will not mind us not being nice to them also. But in social sciences, I think that is where the question is coming from. It really depends, because like I said, there may be some groups of people who will not come forward to answer your question. So you have to be extremely neutral, non-political, and make up your questions so not to kind of offend any kind of, so being politically correct in asking the question becomes very, very important in those cases. So yes, it can happen that you are asking a question and the behavior changes, and I have seen it happening, especially when you're talking about something that they don't want to, information that they don't want to share, especially some sensitive areas, and if you're there. But that is okay. I think you just need to kind of uh, be politically correct when you're asking those questions. some
And
some
that's the job of a researcher to
that
minimize
is,
this effect.
yes, that is the job of the researcher, and that is where the expertise comes. What kind of questions to ask,
Yes.
what kind of so that you get your data, but you are not like changing the behavior, so to speak. And you are able to get that data, because everybody who goes to field to collect the data has very limited time and money available. So obviously, like, you want to finish your work. So that is where.
लास्ट में दो क्वेश्चन हो रहे हैं. एक तो है मैम, कैन यू प्लीज शेयर दिस पीपीटी ऑफ टुडेज मीटिंग?
Okay,
Okay,
I can do that.
although it's not required because we have all directly uploaded the YouTube.
YouTube. Okay.
Yes,
Okay.
so, but it's up to you, ma'am, whether you want to share it or not. So we will let our
There's
person.
another question I see. You mentioned that results differ on taking different sample sizes. How to calculate the correct sample size so that our data is most accurate? Yes, Nisha, I think I had given you the formula in one of my slides. If you want, I can go back to that slide. But there is a formula which tells you how many sample, what is should be your sample size. Sometimes there are ethical issues when you're working with animals. You kind of get away with like rats I was showing. I can get away with only 6 rats for a particular set. Okay, 6 rats. If you show your results, it's fine, because obviously animal ethics says that you cannot use more just for making your data look pretty. Depending on, so plants we don't mind. We grow seedlings randomly. We will sacrifice 100 seedling, take out its protein. It's fine with them. But for animal research, it is obviously limited. Even for human, obviously, like, you get for comparison, sometimes you get disease samples more than the healthy sample, because healthy, nobody wants to give your lung or liver for the research, right? So such kind of things happen, and that is why you have different sampling techniques, and you talk about them and how you have sampled. So that sample size does affect, but there are ways to measure, calculate what is the okay, what is the difference between stratified sampling and quota? So stratified is when you're designing, and within the strata, then you are doing it randomly. Quota is completely like you have designed and you are going to just take from there. So that is more. Megha is asking how many sets of sample enough to order to represent the like, again, like I said, there is a
Just a
formula
second,
that I
ma'am, just a
showed.
second. May I request the participant to mute yourself before so that we can hear? I think, yeah, thank you. Now you can, ma'am.
I was okay. So this basically is the formula which is used. Okay, and so you can calculate what is your population size. Again, what sample you are taking will depend on your population size, right? And population is always, you can define what population you are taking. So if I just want to take Miranda House, one college as my population, numbers are different, right? I have maybe 6,000, 5,000 to 6,000 students. What is the number that I require from there? Okay. But if I take Delhi, the numbers are larger, so your sample size also has to be large. Okay. The formula I have again like put there. So there are formulas which have been tested and vetted statistically, which are there, like which we use. But depending on, first, like I said, what kind of sample, and second, your time and effort and money, everything is considered, of course, but there is a formula which can be used to kind of come to that conclusion, what will be enough. Okay.
Thank you, ma'am. And sorry, participant, we cannot take more queries or questions because of the time constraint. But you can put your question under the ma'am's video on YouTube channel, and either ma'am will directly reply over there, or we will take your question for in the next session. ठीक है ना, ma'am?
Okay. And they can write to me also. There's
Yes.
my email was given, so I suppose you can.
Yes, you can directly write to ma'am. Yes.
That I'm okay with that. Thank you all the organizers. Actually, it was wonderful, and I hope I have been able to answer all the questions.
Yes, ma'am. Yes.
Thank you.
And the number of questions are coming shows that how interacting this session was. Like normally, if there are more questions, it means our participants are directly linking themselves with your presentation, and more queries means more understanding, and it was totally showing that engagement you have through your presentation. Dr. Ishan, would you like to take some time, or I should start the vote of thanks?
I think you can start the vote of thanks. I just wanted to convey that they can use World Bank data. This is a very ultimate and free source of data which provides
Yes.
information not for only means it provides a global data means for all the countries if you want to
Yes.
access data about population, forest cover, agriculture, poverty, so many um hot topics that you want to look for. It provides a very wonderful source and it provides a geographical distribution also for different years. So you can have even visualization also using that software. That is what I
Yes.
wanted to convey.
Mm-hmm.
Yeah, World Bank is a very good source, and because World Bank is a dedicated platform for this kind of data, I also have some suggestion like FAO, Food and Agriculture
Yes.
Organization, that can also be used to collect the data.
Lot of UN bodies have their own sets of data available,
Yes,
which you can basically depend on what you're doing. What I was saying is more science based, but depending on what you're doing, even meteorological data, meteorological offices will kind of share it for free. So if you want to kind of study rainfall or some other temperature and things like that, that are available. So yes.
yes. So there are lots of data, and we have to work on it to get the meaningful information from that. So now it's time for vote of thanks, ma'am. If you will permit us, so can we
No.
formally
Yes.
start? Yes.
You don't
On
have
behalf
to
of the
actually.
organizing committee, I take this opportunity to express my heartfelt gratitude to all those who made this event a success. First and foremost, our sincere thanks to Dr. Somdutta Roy for gracing the occasion with her presence and sharing such valuable insight. We are truly honored. I extend our deep appreciation to our principal sir, Professor Narendra Singh, whose guidance and constant support made this program possible. To the IQAC coordinator, Professor P.K. Sisodia, and the bursar of the college, Professor Mukesh Jain, thank you for your constant support and guidance. I also acknowledge the constant effort of the convener SDCC, Dr. Saima, and fellow colleagues, Professor Dheeraj Singh, who ensured the smooth conduct of the session. A special thanks to all the participants who have registered and shown such enthusiastic engagement. Your presence is what gives meaning to this event. We are unfortunately have the limitation of 100 participants, but I hope that you also connected through YouTube if you are unable to join directly through the meeting. Last but not the least, thank you all once again for being with us. We look forward to continued learning, collaboration, and success throughout the course. Thank you, ma'am. And before we leave, there is one more announcement. In the last week, feedback assessment form, although we have received almost more than 130, but there are still around 10 forms are pending. So please fill it by today's midnight so that we can release your marks accordingly. And make sure that your name and your affiliation are properly mentioned so that because the same will be reflected on your certificate. And now you can also mark your attendance on YouTube and on this platform to ensure your certificate. Thank you, everyone. Thank you so much. And one more
I
Thank
time,
would
you.
like to add one more thing, sir.
yes, sir.
The answer for this quiz will be released after the forms are closed, right, sir?
Yeah, yes, sir. Yes, sir.
So that they can check their answers. Thank you, everyone. The meeting is over. Now you can leave the meeting.
After marking your attendance.